Oh, you're good. Okay.
Suddenly, I'm so nervous. Okay.
We are recording now.
Hi, I'm Anaí de la Vega, and the title to our project is The Evolution of Pelvic Spines in 3 Stickleback Populations, and the names of my partners are
Tyler Michelle
and Tyra White.
The outline of our presentation is going to be as follows. We're going to explain the materials that are used in the experiment. We're going to give insightful details with the background information and give context of our experiment. We're going to discuss the question and hypothesis and explain the null and alternative hypothesis. We're going to be able to state the dependent and independent variables. We're going to explain the procedures that we took to conduct this experiment. Finally, we're going to be able to analyze our results using charts and graphs and go over the conclusion, where we will accept or reject the alternative and null hypothesis. And finally, the discussion of broader impacts will undergo.
Okay, our materials for this project is going to be, of course, gloves, 20 fish from Bear Paw Lake, and 20 from Frog Lake. We're going to be having a strainer, Alcerian red staining solution, a waste container, destaining solution, 50% isoprene alcohol solution, and a magnifier that will be used for this whole entire project.
Okay, so I'm going to go ahead and get started about the background. It's a seafaring stickleback fish that supports a pair of spines that sticks out, and it helps the fins. The freshwater cousins look far less of being threatened and outfitted of a much smaller spin. The researchers have found a lot of gene activities that have been going on. It's been rare demonstrations how it's a small genetic change that rapidly has an impact of an organism. It all started with the saltwater fish about 10,000 years ago. Some of the fish were just trapped inside of a lake, inside the glacier ice. And during the overtime of the lake fish, it came to look quite different. So some of these marine forefathers have different types of genes and different activities that's going on. It's more of like a population of the laboratories for the evolution studies. The marine fish have protein spines. Some of them have different spines. They're not all the same. Their new environment was short of the minerals needed to make the spine. It consumed too much of the fish's energy to be the worthwhile. For the breeding saltwater, somebody had the study from Stanford, and some of his friends decided to track down the loss of the spines. Also showing the spine, the similarity of the spines, they didn't appear when they did their project, so it all went down because it had the lack of bad materials that were going on. And yeah, so they influenced the gene that was called the PIX1. It had expressed many of the developments that was going on. It was found as an active pelvic girdle of the marine sticklebacks, but it was not a freshwater fish. So the PIX1 had a lot of activity going on, but it wasn't suggested with the freshwater fish. It had changed the genes of a regulation, so it was the gene itself. The lake sticklebacks had lost their own spines in April the 15th, and it was due to the issue of the nature. The activity was smaller than normal, hid to the binds. It was symmetrical to the freshwater stickleback. And then many studies have shown that it is important because it changes the profound of the effects.
So the question we are going to be trying to prove is that are the two different locations and contributing factors to each location, Bear Paw and Frog Lake, playing a role in the pelvic structures in the stickleback fish. Our hypothesis consists of the null and alternative hypothesis. Our null is the fish in Bear Paw Lake will see more alterations in pelvic bone structure than the changes seen in the fish from Frog Lake. Our alternative to that hypothesis is that the fish in Bear Paw Lake will see less structural changes in their pelvic bones than changes seen in the fish from Frog Lake. Our dependent and independent variables are the variety of fish that are being tested in the two different lakes to see if there will be changes in the pelvic structure. That's our dependent variable because that's the variable that's going to be affected in the experiment. The independent variable are the two lakes that the fish are going to be living in, Bear Paw and Frog Lake, because these are going to be contributing any alterations in the pelvic bones.
Okay, starting off with the procedure, we'll of course be getting our gloves, and we'll be getting the 20 fish from Bear Paw Lake and 20 fish from Frog Lake. And basically these fish were brought out from our scientists who trapped the fish in a fish trap and was able to bring it to us in our lab for today. So with these fish, we're gonna get them from their jar, and this is gonna be for both of the 20 from Bear Paw Lake and 20 from the Frog Lake. It's just we're doing it in separate little transitions. So first we'll get the alcohol and pour it into the jar to basically strain them, and this prevents them from falling out of the jar. After that, we'll be getting the Alzarian staining solution, which stains the fish red, and we need this in order to see the actual fish when under the magnifier, just to see if the fish is reduced, complete, or absent. After this, we'll have to wait 3 to 6 hours for it to actually stain the fish, which is very important because it needs to be in order to be that long so it can actually stain the fish. And then after that, we'll pour it into the waste container. And then we'll get the destaining solution, so it kind of washes out the fish, but it stains the actual bone, so we can see that part. So it's not confusing when we're looking under the magnifier. After that, we're gonna be doing this rinsing method where we add water and then we shake it a little bit, and then we wait like 12 hours, and we have to do this several times of rinsing the fish to make sure it's completely rinsed. After that, we have to get the fish, we pour it into an empty tray, and we start analyzing each fish individually. In the picture that you see here, you can see that this is a complete sickleback fish, and this is like what we go off by, and we'll just write it down in our results and be like, this is complete, this is absent, this is reduced, and we'll keep on doing that method. We did do a little experiment on the fossils, which we did get some rocks from it, and we did do some analyzations from that. But since we are mainly focusing on just Bear Paw Lake and Frog Lake, we're not really mentioning it that much. Okay, with our results, in the next slide, you'll see our graph, and it does show that Bear Paw Lake did have an absent or reduced pelvic phenotype, which basically means that they don't have pelvic spines. And all the fish from Frog Lake did have complete pelvic spines. So with this, it means that in Bear Paw Lake, they don't really specifically have predatory fish, because in order for them to have the pelvic spine to be actually significant to them is to fight against predators against them. And again, from me explaining briefly about the fossil fish, in the line graph, you'll see that through the years after the colonization of the lake populations, it'll show how it went from the population of their complete pelvis spine, which you can see on the thing, where you can see in the 3,000 and 6,000 where it dropped completely. And then in our other ones, you can see in Bear Paw Lake that it had 3 complete, 10 reduced, 7 absent. And then in Frog Lake, where we completely had 19 of the fishes being complete and 1 reduced, which means basically all of them are complete. So in conclusion, our hypothesis of the fish in Bear Paw Lake will see more alterations in the pelvic bone structure than the changes seen in the fish from Frog Lake was correct. pretty much, again, it shows that Bear Pollock doesn't have really that many predators, and that's why their evolution of them not having it shows in that little section. And compared to Frog Lake, where they have predatory fish like the trout, and it's just been ancestral for them since the beginning of the time, where we could see through the fossils.
So for broader impacts, we first have to discuss the possible group making in order to further this research. Something different, like some that have similar pelvic structures or even similar protective structures, to see if there are similar evolutionary changes like the stickleback population. Can you guys see that? Mm-hmm. We also see that there's different variables to experiment in this experiment. We can add different fish that could be predatory in the group or the lake that doesn't have predatory fish, and see if there are major changes in the experiment. As in evolution, we see that there are predatory contributors which cause a vestigial trait in the lake that doesn't have predatory contributors, and this is because it's no longer needed, whereas in Frog Lake, they would need the predatory or the protective structures as the pelvic structures. In this graph, we can also see a directional selection that is due to lack of mating between the species of stickleback fish, which this usually causes two types of species in this population. We see that at 0 years, they have 100% pelvis structure, and it drops to 80 at 3,000 and then below 20% at 6,000. So we obviously see less and less with pelvis structures. And finally, we also see that there is natural selection due to predatory contributors. The fish that need protection will have the pelvis structures, and vice versa. If they don't need the pelvis structures, then they probably don't live in a place with a lot of predatory contributors.
And then here are some of the references we did use throughout our project.